Imagine a world where we can rewrite the code of life, fix genetic defects, cure diseases, and even improve human abilities. This is the promise of CRISPR technology, a revolutionary tool that has taken the scientific community by storm. CRISPR, short for clustered, regularly interspaced short palindromic repeats, allows scientists to edit DNA with unprecedented precision and ease. This groundbreaking technology has the potential to change medicine, agriculture, and even how we understand life itself. Before CRISPR, gene editing was a long, cumbersome, and imprecise process. Traditional gene therapy methods often relied on viral vectors to deliver genes into cells, which could lead to unpredictable and potentially harmful results. CRISPR, on the other hand, acts like a pair of molecular scissors, allowing scientists to target and cut specific DNA sequences with incredible accuracy. The discovery of CRISPR has ignited a wave of research and development, with scientists around the world exploring its vast potential. From developing new treatments for genetic diseases to genetically engineering pest-resistant crops, CRISPR has opened up a world of possibilities. However, this new power also comes with profound ethical considerations. As with any revolutionary technology, it is important that we proceed with caution, carefully weighing the potential benefits against the risks. The ability to edit the building blocks of life raises fundamental questions about human nature, the limits of scientific intervention, and the kind of future we want to create. CRISPR technology, despite its revolutionary applications, actually stems from humble beginnings. It was first discovered in bacteria, where it serves as an adaptive immune system, protecting these single-celled organisms from invading viruses. Bacteria, despite their simplicity, face constant threats from viruses which can infect and destroy them. CRISPR evolved as a way for bacteria to remember and defend against these viral invaders. The CRISPR system in bacteria consists of two main components: CRISPR sequences and CRISPR-associated CAs enzymes. CRISPR sequences are unique segments of DNA that act as a genetic library of past viral infections. These sequences contain short snippets of viral DNA called spacers interspersed between repeating DNA sequences. CAs enzymes, on the other hand, are the molecular scissors that provide the cutting action. These enzymes work together with CRISPR sequences to target and destroy invading viral DNA. When a virus infects a bacterium, the CRISPR system captures a small piece of the viral DNA and integrates it into the bacterium's genome as a new spacer within the CRISPR array. This process of acquiring and storing viral DNA fragments provides the bacterium with a genetic memory of past infections. The CRISPR system can then use this information to recognize and destroy the same type of virus if it encounters it again. The CRISPR mechanism in bacteria is a fascinating example of how nature has developed elegant solutions to complex problems. The process begins with the transcription of CRISPR sequences into RNA molecules. These RNA molecules, called CRISPR RNAs, cRNAs, act as guides, directing the CAs enzymes to their targets. The cRNAs bind to the CAs enzymes, forming an active protein RNA complex that can recognize and bind to specific DNA sequences. The complex then scans the cell for any DNA that matches the sequence encoded in the cRNA. When a match is found, the CAs enzyme cuts the DNA, effectively disabling the invading virus. Scientists realized that this bacterial defense system could be harnessed for genome editing by designing synthetic cRNAs that target specific DNA sequences in any organism. They could direct CAs enzymes to cut DNA at precise locations. This breakthrough opened up a world of possibilities for manipulating genes. After the DNA is cut, the cell's natural DNA repair mechanisms kick in. These mechanisms can be exploited to introduce changes in the genetic code. CRISPR technology holds immense promise for revolutionizing medicine and addressing some of the world's most pressing health challenges. Its ability to edit genes with high precision opens new doors for treating and potentially curing genetic diseases previously considered incurable. One of the most promising applications of CRISPR is in the field of gene therapy. By correcting faulty genes that cause disease, CRISPR offers hope to patients suffering from conditions like cystic fibrosis, sickle cell anemia, and Huntington's disease. Clinical trials are already underway, testing the safety and effectiveness of CRISPR-based therapies for various genetic disorders. Beyond treating inherited diseases, CRISPR has the potential to revolutionize cancer treatment. Scientists are exploring ways to use CRISPR to engineer immune cells that can more effectively identify and destroy cancer cells. This approach, known as CAR T-cell therapy, has already shown remarkable success in treating certain types of leukemia and lymphoma. CRISPR's applications extend far beyond human health. In agriculture, CRISPR is being used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. This can lead to increased food production and reduced reliance on pesticides, benefiting both the environment and human health. While the potential of CRISPR is enormous, it's crucial to acknowledge the ethical considerations and potential risks associated with this powerful technology. The possibility of off-target effects, where the CRISPR system mistakenly cuts DNA at unintended locations, is a concern.